Good morning, friends. Good morning, students. I am back with another video. Today we will discuss urea cycle. So first of all, what is urea? Urea is end product of amino acid metabolism, right? So the other names of urea cycle are Krebs, Henseleit cycle. The second, there is also another name that is also known as ornithine cycle, right? And so this is the structure of urea. Two nitrogens are there, one carbon is there. The carbon is derived from carbon dioxide. The carbon dioxide is in the form of bicarbonate, right? And one amino is derived from ammonia, right? And one amino group is derived from aspartate, right? Now, remember the difference in multiple choice questions. If only Krebs cycle is written, then it is TCA cycle. If Krebs Henseleit cycle is written, it is urea cycle, right? Or otherwise, there will be only mentioning ornithine cycle, right? Now coming on to the organ. Where does it take place? It takes place in liver, right? And organelles, there are two organelles. The first two steps take place in mitochondria, right? The next steps, third, fourth, and fifth, take place in cytosol, right? Now we'll start with urea cycle proper. Now, this is proper urea cycle, and the first step is carbon dioxide and ammonium ion. They combine with each other to form carbamyl phosphate, right? And the enzyme involved is carbamyl phosphate synthetase 1, right? There are two enzymes in the human system, that is carbamyl phosphate synthetase 1 and 2. So we'll start with their differences. The carbamyl phosphate synthetase 1 is involved in urea cycle, right? Here, right here. And carbamyl phosphate synthetase 2 is involved in pyrimidine synthesis, right? And it is a mitochondrial enzyme. Carbamyl phosphate synthetase 1 is a mitochondrial enzyme. And CPS2 is a cytosolic enzyme, right? And this enzyme requires an acetyl glutamate as a cofactor, and CPS2 does not require any cofactor, right? And the nitrogen is derived from ammonia, right? And in this case, it is derived from glutamine, right? Now, as we discussed, carbon dioxide ammonia in the presence of carbamyl phosphate synthetase 1, it is converted to carbamyl phosphate, right? So this is the first step, and the first two steps take place in mitochondria, right? So we can divide the urea cycle here. This steps beyond this line, this is mitochondria, right? And this is cytosol, right? Beyond this is mitochondria. Beyond this is cytosol, right? So in order to remember the five steps, you can use the mnemonic CPCASAO, right? C for CP for carbamyl phosphate, then C for citrulline, then AS for adenosine, then A for arginine, and O for ornithine, right? So this is the ornithine is the second last step in the completion of this cycle, and it can be compared with TCA cycle, oxaloacetic acid, right? In case of TCA cycle, the oxaloacetic acid is the second last step, and in this case of urea cycle, ornithine is the second last step. Now, as we discussed, carbon dioxide derived from bicarbonate and ammonium ion combined with to form carbamyl phosphate, right? This is the first step, and this takes place in mitochondria. Then the second last step product that was formed was ornithine. Then ornithine and carbamyl phosphate in the presence of ornithine trans carbomylase, right? Ornithine, the second last product, combines with carbamyl phosphate in the presence of enzyme ornithine trans carbomylase to form citrulline, right? Citrulline, this is the second step, right? Citrulline then forms adenosine by combining with aspartate, right? The enzyme involved is adenosine synthetase, right? And there is utilization of ATP, 2 ATP here, and we get AMP, right? And the third step is arginosuccinate, and this takes place in cytosol, as we discussed earlier. Then arginosuccinate will form arginine, and arginosuccinase enzyme is involved, right? Now we got fuminate here. Fuminate can enter TCA cycle and also involved in gluconeogenesis, right? After arginosuccinate, we get arginine. The fourth step and enzyme involved is arginosuccinase, right? Arginine will undergo hydroxylation reaction to form urea by the enzyme arginase, right? And we'll finally get fifth product, that is ornithine, right? And ornithine here is regenerated, right? So it is regenerated, it will further combine with carbamyl phosphate and urea cycle will proceed, right? So the 5 steps were major. In order to remember the code CPCASAO, CP for carbamyl phosphate, C for citrulline, AS for arginosuccinate, A for arginine, and O for ornithine, right? The 5 steps were there, and there is net utilization of 4 ATP in the synthesis of urea, right? So first, here in the first step, when carbon dioxide and ammonium ion combine, the 2 ATP are utilized. And in the second and third step, during the synthesis of arginosuccinate, here we consume 2 ATP to form AMP and phosphate, and we get arginosuccinate, right? So now we'll start with regulation of urea cycle, right? The first step is very important, regulation. The carbamyl phosphate synthetase 1 reaction. So this is rate limiting step, right? And CPS1 is directly controlled by cofactor, that is N-acetyl glutamate, right? And N-acetyl glutamate is directly proportional to the concentration of arginine, right? So when we eat protein rich meal, the concentration of N-acetyl glutamate will increase, and it will further stimulate carbonyl phosphate synthesis 1, and urea cycle will keep on proceeding, right? And the rest 4 enzymes, second, third, fourth, and fifth, they are controlled by their concentration, relative concentration of their respective substrates. Now we will start with disposal of urea from the body. So we already know that urea is produced in the liver from the previous section. Now the urea produced in the liver is transported to kidneys, right? And from here it is excreted out of the body, right? And small amount, very small amount, may also enter intestine, right? From here, it enzyme urease acts over the urea to form ammonia and carbon dioxide, right? So if small amount enters the intestine, it will be acted upon by urease enzyme to form ammonia and carbon dioxide. And please keep on studying and do like and subscribe the channel. If you have any queries, you can message in the comment section below. I will try to make videos over it as soon as possible. Thanks a lot. Best wishes.